Good afternoon, everyone, and welcome to today's webinar, Prebiotics and Mood, Exploring the Gut-Brain Axis. I'm Leslie Samay, Director of Professional Development at Great Valley Publishing Company, and I'm your host for today's show. Before we start our presentation, I have a few points of housekeeping. First, in order to claim your credit, you will have to remain with us through the entire 60-minute presentation. Second, we'll address questions at the conclusion of the webinar. If you use the Q&A box to send us your questions, we'll try to get to as many as we can. And next, Dr. Robert Rastall, PhD, has the following relevant disclosures to report. He's received research funding and/or speaker honoraria from Benio Institute and Prebiotics Health PLC. Funding from non-CPE revenue for CPE planning, development, review, and/or presentation has been provided by Benio Institute. And the responsible provider for this program is Benio Institute. And Prebiotics and Mood, Exploring the Gut-Brain Axis awards 1 CPEU in accordance with the Commission on Dietetic Registration's CPEU Prior Approval Program. And now I'm happy to introduce today's presenter. Dr. Bob Rastall is Senior Tutor within the Department of Food and Nutritional Sciences at the University of Reading in the UK, where he also holds the post of Professor of Food Biotechnology, teaching food chemistry and biochemistry. Dr. Rastall has a BSc in Applied Biology and a PhD in Microbial Biochemistry from the University of Greenwich, London. Professor Rastall's research interests are around developing novel enzymatic manufacturing technologies for functional carbohydrates targeted at gut health. And now I'm pleased to turn it over to Dr. Rastall. Welcome.
Thank you very much, Leslie. Well, in the interests of not distracting you, I'm going to turn my camera off. And I'll turn it back on again at the end when we do some questions. So the learning objectives are to define the term prebiotic, and we will look at what a prebiotic is and what a prebiotic isn't. We'll discuss the impact of prebiotic consumption on the gut microbiome. We'll explain the evidence that supports the impact of prebiotics on the gut microbiome, and hopefully allow you to counsel patients and clients on the impact that consuming prebiotics might have on anxiety and depression. So let's start off by answering a question I often get asked by people, and that's what's the difference between a dietary fibre and a prebiotic? And there's a lot of confusion over this. The two things are actually related. If we look on the left-hand side of this slide, we see some carbohydrates there: resistant starch, type 1, cellulose, curdlan, chitosan, chitin. These are not really fermented to any great degree by the gut microbiome. Over towards the right, we have carbohydrates that are much more readily fermented. Galacto-oligosaccharides, oligofructose, inulin. We'll talk more about these carbohydrates shortly. But as we go across that slide, we increase the fermentability, we increase the solubility, but not all of those carbohydrates have the same effect on the microbiome. As we increase solubility, we have an increase in bacterial population. The soluble carbohydrates there are substrates for bacteria, and we will increase the population of bacteria in the gut using these materials. But they're not all built the same way. They have different structures. And the precise pattern of bacteria that we grow using these carbohydrates depends on the structure of the carbohydrate. So not all soluble fibers are the same. That bacterial population that we're developing using those soluble fibers will produce metabolites, will produce gases, will produce short-chain fatty acids, and we'll say more about those as we go on, because those are important mediators of the health benefits of prebiotics. And again, the structure of the carbohydrate can influence the pattern of those metabolites. Well, we better define what we mean by a prebiotic. Now, there are quite a few definitions of prebiotics kicking around. This is the one that I use and the one that I would urge you to use. This is the latest consensus definition from the International Scientific Association for Probiotics and Prebiotics. So this was a consensus definition by a group of experts in the field, industry experts, academic experts, And they debated this definition. The definition they came up with is this one: a substrate that is selectively utilized by host microorganisms that confers a health benefit. Now, I've highlighted two phrases in here that we need to think about: selectively utilized and health benefit. So selectively utilized means that health positive organisms in the gut are using this prebiotic as a substrate. As a consequence, we're getting some kind of health benefit to the host. Now, it's important we bear in mind these two components of the definition. And the reason is that showing selective utilization by host microorganisms is very easy. Conferring a health benefit, showing that is a lot harder. And there is a trend for people to show the first half of this definition and then start using the word prebiotic when they haven't actually demonstrated that they have a health benefit. So I'd like to stress really that we need to establish both of these in order to use the term prebiotic. Following on from that definition, you can see the reference to the paper at the bottom of the slide there. This is a diagram that I've adapted from that paper. We can really think about substances that enter the gut, and we can divide them up into ones that have selective utilization by microorganisms and others that will affect the microbiome but are not substrates for bacteria. So, for example, we can think of prebiotics and not prebiotics. Substances that affect the microbiome could be either. Selective utilization by microorganisms could be a prebiotic. So prebiotics might, for example, be oligosaccharides such as galacto-oligosaccharides, fructo-oligosaccharides, inulins. Potentially could be CLAs and PUFAs. They could be human milk oligosaccharides. We'll talk a bit more about that later. They could also be phenolics and phytochemicals. There's not really any rule that says a prebiotic has to be a carbohydrate. All of the currently accepted ones are carbohydrates. They're the ones where we actually have the definition properly fulfilled. On the right-hand side in red, we've got things that aren't prebiotics. Probiotics are not prebiotics. Proteins and fats we wouldn't normally consider as prebiotics. Vitamins, antibiotics, they will affect the microbiome, but they're not prebiotics. In the middle, we've got this grey area, actually it's a blue area, dietary fibre. So they can be readily fermentable, as we've seen they might be prebiotic, or they could be less fermentable, in which case they might not be. So with the dietary fibre, it really depends on the molecular weight and the structure, whether they are prebiotics or not. Now, this is my very simplistic diagram of the gut microbiome. This barely scratches the surface, really doesn't scratch the surface of the complexity of the bacterial ecosystem that we have in our guts. But what this does do is indicate some known functional groups that we know are in there and some of the ecological interactions between them. So at the top, we have sources of nutrition into the colon. So we have oligosaccharides, we have polysaccharides, mucins, proteins and peptides. These will stimulate the population and activities of certain organisms. The ones outlined in green are ones that have been repeatedly associated with health. The ones in the red box there have been associated with poor health, specific disease conditions. The ones in blue, we don't really have a very strong association, health positive or health negative, but they are critical components of the gut microbiome. If we look at the pathway from oligosaccharides through Bifidobacterium and lactobacillus down to acetate, we can see that the Bifidobacterium and lactobacilli directly produce acetic acid. They don't produce butyric acid, butyrate. But the lactic acid and the acetic acid can be used by other organisms, such as the Clostridium 14A group, to produce butyrate. So we have this complex network of cross-feeding reactions that occur in the colon. And it's important that we bear this in mind when we're thinking about prebiotics. We are not talking about drugs. We're not talking about one molecule hitting a physiological target and producing a health benefit. We're talking about changing the ecology of a complex ecosystem. And it involves changes in many different organisms to produce changes in metabolites. And we'll see some data when we look at the study that I want to present to you in a moment. At the bottom of this slide, on the left-hand side in green, we have acetate, butyrate, propionate. These are short-chain fatty acids. These are all health positive. And these are mediators of various physiological processes. I'll say more about that in a moment. And these are generally associated with health. In the red boxes on the right-hand side, we have various compounds which are probably toxic in the gut. They are certainly toxic in vitro, and they're toxic in animal models. We don't really know for certain that they are toxic in the gut because we don't know what concentrations they're produced in the gut. But it seems like a good idea to minimize their quantities and increase the quantity of the short-chain fatty acids. So really, prebiotics are designed to shift this equilibrium of this ecosystem over towards producing those health positive metabolites. Another component of being a prebiotic, of course, is that it needs to reach the colon. If it's metabolized by your small intestine, it's going to have very minimal impact on your gut microbiome. Now, we do know quite a lot about the brush border enzymes in the small intestine. There are several enzymes that we know about. These are very, very adept at metabolizing certain oligosaccharides, particularly glucooligosaccharides. If we look at the specificity constants there for these enzymes, however, it's obvious that the structure has an impact. And some oligosaccharide structures are very readily metabolized by the small intestine, others less so. 1,4-linked glucose oligosaccharides very readily absorbed. 1,6-linked and 1,2-linked much less so. Generally speaking, though, alpha-linked glucose containing carbohydrates are fairly well handled by the gut. Beta-galactosidase is also present in people that are not lactose intolerant, and that enzyme will metabolize lactose plus other galacto-oligosaccharides. This means that some components, not all, but some components of commercial galacto-oligosaccharide products are susceptible to hydrolysis by those beta-galactosidase enzymes. So when considering if a component is prebiotic or not, apart from the definition that we've already shown you, you need to think about absorption. Is it actually going to get through the small intestine and into the colon? Now, there are an awful lot of materials out there where people are using the word prebiotic. And it's very, very easy, as I said, to do some microbiology on a carbohydrate and show that health positive bacteria are increased. That's not really enough. We do need to show those health benefits in people. So this is my view, it's a personal view of how we are at this moment in time. In the red, we have candidates. So these are all carbohydrates where we have some evidence of manipulation of the microbiome. All of these will change the microbiome in a positive direction, if you like. They will all have increases in health positive bacteria in vitro. That is not the same as showing that that works in vivo. In the orange there are candidates with some good data, but generally we're lacking compelling evidence for actual health benefits in humans. Quite often there are studies that will establish changes in the microbiome using fecal analysis, for example, but without the health benefits, those health studies are not easy to do. They're very expensive and quite time consuming. There's a lot of effort involved, and not everybody has funded those studies. In the middle, we see inulin, oligofructose and GOS in the green circle there. Those are the ones that I would consider to be the established prebiotics. And we have good evidence for impact on the microbiome, both in vitro and in vivo. And we also have evidence of health benefits, a range of health benefits. So those three really, I think, at the current time, are the established prebiotics. Now, I expect over time we will see a migration of these compounds from the outside of this diagram towards the middle. So I would expect that green circle there to become a bit more populated in the future. We've talked a little bit about testing for prebiotics. I think it's worth thinking about what evidence we need in order to make this claim. I always see a hierarchy of evidence and a hierarchy of understanding, and the two kind of go together really to come to a prebiotic claim. Over on the left, the simplest way of evaluating carbohydrates and their effect on the microbiome is in pure cultures. You simply take a pure culture of a bacterial strain in a Hungate tube, see whether it grows on a given carbohydrate. These are useful to understand the physiology of a microorganism's interaction with a candidate prebiotic. And we need to do this kind of work to understand the impact on microbial physiology. But it's a long way short of what we need to come up with the claim that it's a prebiotic. We can then use co-cultures. And these again are really useful for understanding microbial behavior and probing the function of that ecosystem. In vitro gut models, a more complex way of investigating the gut microbiome. And again, these are really, really useful for understanding the ecosystem and understanding microbial behavior. But it doesn't tell you necessarily what will happen in a human. Animal studies have their place, particularly when it comes to looking at health. There are various animal models of disease which can be used, and these contribute to understanding disease mechanisms. We don't really have any good animal models of humans when it comes to the gut microbiome, however. So animal studies can't really tell us whether something's going to be a prebiotic in people. In order to do that, we need to do human volunteer trials over on the right-hand side of that diagram. Then we can make a prebiotic claim. And I do think we need to have some rigor over how we use this term prebiotic. I can understand why marketing people in companies would like to use this term, but as a scientist, it does frustrate me somewhat when I look at the evidence and I can't see it. So I think I would like people to have a bit more rigor and a bit more critical thinking when it comes to using this term. Okay, so what happens when you consume a prebiotic? Well, this is a diagram that has evolved over many years, and it will carry on evolving. I keep having to make elements smaller to fit them on the slide. But this really summarizes plausible mechanisms connecting prebiotic fermentation to health. I'm not going to go into great detail on all of these. All of these mechanisms that are on this slide have some evidence behind them. They're all plausible mechanisms with some element of evidential support. Quite often this support comes from gut models or from animal models. It's very difficult to establish in real live human beings this connection between metabolism in the gut and health. We don't have realistic access to people's guts. All we can really do usually is to collect fecal samples and use feces as a surrogate for what might be happening inside the colon. What we can see here is that consuming prebiotics, will bring about changes in the gut microbiome, and that will bring about changes in the metabolites that are formed. We have our short-chain fatty acids that I introduced you to earlier. These will have a variety of impacts. But we also see the production of neurotransmitters. So we have gamma-aminobutyrates, dopamine, norepinephrine, serotonin, histamine. These are potentially active on the brain, and they are part of the gut-brain axis. The brain and the gut are linked with neuronal connections, two-way connections using the nervous system. But these metabolites, we believe, are also key components of this system. It's not absolutely certain at the moment that these neurotransmitters can cross the blood-brain barrier. So there is still some doubt over the importance of these neurotransmitters. But as we'll see later, consumption of prebiotics can indeed affect the brain. So it's this gut-brain axis that I want to focus on really for the rest of this talk. In particular, I want to look at improvements in mood. There have been some studies on prebiotics in mood, not really very many, to be honest. Anxiety and depression are the two main outcomes that have been studied. We can see them on this diagram here. And if you look at this, you can see clearly that some studies have shown an effect and some studies haven't. It's rather mixed picture, really. We don't really have any clarity from the literature over whether there are any effects. Also, when people do meta-analyses like this, they have a habit of looking at prebiotics generically. And you can see that's what's happened here. We have short-chain FOS, oligofructose, galacto-oligosaccharides, and inulin oligofructose mixture. These are all different, and they would not necessarily be expected to have the same impact. They will have differential effects in the gut that produce different metabolites. Why would you expect them all to have the same effect on the brain? And this is a bit of an issue when we're looking at meta-analyses involving prebiotics, and something you need to look out for. People will sometimes make a claim that a prebiotic doesn't have evidential support, and when you look at it, they've used 3 different agents. So we set out to do our own study. This was funded by Benio. We got this published in the American Journal of Clinical Nutrition. We're very, very pleased to get this paper in there. Now, this work was actually carried out in the lab by my wonderful former PhD student, Peter Jackson. When people like me talk about my research and my research interests, in reality, it's the efforts of our PhD students and postdocs. And I want to credit Peter Jackson here because he was a superb PhD student. He did a fantastic job on this study. Well, we set out to see whether we could alleviate anxiety and depression in a group of volunteers. So this was the study design. The interventions that we looked at were oligofructose, 2-fucosyllactose. This is a human milk oligosaccharide. It's being added to infant formula, and there's increasing interest in looking at whether or not adults can benefit from intake of these human milk oligosaccharides. We also looked at a blend of oligofructose and 2-fucosyllactose. We had maltodextrin as a placebo. The inclusion criteria, we wanted 18 to 50-year-old non-smokers. We screened them using health questionnaires. So we gave them the PHQ-9 and GAD-7 to look for signs of depression and anxiety. So these are common screening tools. These are the kind of things that you will get readily access to on the internet if you want to check your own symptoms. So we were looking for people with mild to moderate depression and mild to moderate anxiety. We specifically didn't want people that were on medication. We wanted people with at least 3 bowel movements per week. The exclusion criteria, as is usual in this kind of study, probiotics and prebiotics 4 weeks prior to intake. Antibiotic use 4 months prior. Antibiotics have a disastrous effect on the gut, so we have to exclude those. But we specifically excluded antidepressants. And the reason we did this is these antidepressant medications are very, very powerful. And we did not believe that food components would approach what could be achieved with medication. So we excluded them, and we were looking very much at the mild end of anxiety and depression. We also excluded previous psychiatric episodes and also drug and alcohol abuse. This was the study design. We had a pre-screening period. We then assigned the volunteers into 4 groups. 23 volunteers per group. They did daily bowel habit diaries during a 1-week run-in phase. We then went into the intervention phase. So they were fed oligofructose, 8 grams a day, plus maltodextrin at 2 grams a day. Maltodextrin as a placebo, 10 grams. The blend, 8 grams plus 2 grams. That's 8 grams of oligofructose and 8 grams of oligofructose and 2 grams of 2-fucosyllactose. We chose these doses based on previous literature. So during the intervention phase, we took various samples and did various measurements. So we measured as outcomes Beck Depression Inventory. This scores range from 0 to 63. This is a 21-item questionnaire with overall scores associated, higher levels of depression with higher scores. State Trait Anxiety Inventory, STI-Y1 and STI-Y2. These are 20 questionnaires. STI-Y1 is the state trait. It's right now. How do you feel right now? And STI-Y2 is a trait. So generally speaking, over a longer period of time, how anxious are you feeling? These scores range from 20 to 80. We also looked at the positive and negative effect schedule, the PANAS. This assesses transient mood. And there are 20 effect measures, 10 positive and 10 negative. So a lower PA and higher NA are associated with worsened mood. We also looked at the Pittsburgh Sleep Quality Index, PSQI. These scores range from 0 to 21. Now, I'm not actually going to present these because we didn't really get any conclusive data on those. I will show you the other data. We also analysed the effect on the microbiome. So we did some 16S rRNA gene sequencing as a way of probing the effect on the microbiome. We didn't, however, look at relative abundances, which is what a lot of people do. We measured the total bacterial count using fish. And we had bifidobacterial counts using flow cytometry with fluorescent in situ hybridization. And what we do is to normalize the sequencing data against the fish data to get a more quantitative response. I'll show you those data shortly. We also measured urinary metabolites by protein NMR metabonomics, but we didn't really get anything useful from that at all. Unfortunately, we were prevented from doing plasma. And the reason for that is the COVID pandemic. This study was done under COVID restrictions, and our human nutrition unit was closed by the government. We simply couldn't get people into the building and we couldn't take blood samples, which is a shame because what we really wanted to do was to look at plasma and urine and then look at where metabolites might end up. So we did have a compromise there, I'm afraid, that we just had no choice but to accept. Other measures, we measured bowel habit, we measured food intake, so we had food diaries, We also measured salivary cortisol as a measure of anxiety, but again, we didn't really get very consistent data with the cortisol measurement, so I'm not going to present those. So what did we find? If we look at the intestinal microbiology, so this is flow fish, that's fluorescent in situ hybridization. And we can see on the left we got significant increases in total bacteria, the EUB 1, 2, 3 there. These are probes that measure the total bacterial population. So we get significant increases on the oligofructose, on the blend of oligofructose and 2-fucosyllactose, and on the 2-fucosyllactose, the human milk oligosaccharide. And we didn't see any significant change on the maltodextrin. Now, our hypothesis behind this study really was that increasing health positive bacteria will increase the production of neurotransmitters that will have an impact on anxiety and depression. And the group of organisms that we know produce neurotransmitters include Bifidobacteria. So we specifically measured bifidobacterial count, and we can see very strong, significant increases on the oligofructose, on the blend, and also a less pronounced, but still significant increase on the 2-fucosyllactose. We didn't see any increase on the maltodextrin. If you look at the profile of the gut microbiome, so this is sequencing data, remember, normalized to the total count. So we call this quantitative microbiome profiling. We see changes in various groups. The biggest changes were in Bifidobacterium, Bacteroides, and Prevotella, which is not too surprising, to be honest. The profile that we see here is pretty much what we would expect when feeding somebody prebiotics. And you can see the biggest increases in Bifidobacteria there are associated with the oligofructose. The oligofructose 2-fucosyllactose blend, no real difference on the maltodextrin, and a more moderate increase on the human milk oligosaccharide on the right. We look at bowel habits. Again, these results are not too surprising. We know that fructans will improve bowel habit. Here in Europe, there's a EFSA health claim around this. So oligofructose increases stool consistency, so does the blend, but neither of the other two interventions. We didn't really see much effect on stool frequency. There's a slight increase on the blend, but the others really, we didn't see much effect on frequency. This isn't really what we were looking at in this study, however. Much more exciting are the results on the Beck depression inventory. And we saw quite marked decreases in depression in these volunteers. On the oligofructose 2FL blend, and also in the 2-fucosyllactose on the right. So all of our active interventions showed significant reductions in depression, and we had no impact at all with the maltodextrin. Now, what I find interesting is to map onto this the kind of clinical indicators. So 0 to 9 is minimal, 10 to 18 is classed as mild, 19 to 29 is classed as moderate, and 30 to 63 is classed as severe. Now, we screened these people, remember, to have mild to moderate depression. Some of them actually turned out, when we used the Beck depression inventory, a different score. It turned out that they were perhaps a bit more severe than we were originally thinking they would be. Well, I think what's really exciting here is if you look from a clinical perspective, we were taking many individuals from moderate down to mild or minimal. And this, I think, is very, very exciting result. We thought we might see a positive effect. We hoped we might, but we were not expecting to see such a dramatic improvement in the mental health in these volunteers. If you look at the anxiety measures, state trait anxiety, so remember STY-Y1 on the left, that's right now how they felt in the moment. Y2 on the right is how they felt more generally. And again, we see improvements in all of the active interventions. No real improvements at all with the maltodextrin placebo. If we put on the clinical markers again, 20 to 30 is no or low anxiety. Moderate anxiety is 38 to 44 in the orange there, and high anxiety 45 to 80. So again, we were taking people from states of high anxiety down to moderate or low or no anxiety. So we were very, very pleased with these results indeed. I think these have clinical relevance beyond what we were expecting. If we look at the PANAS, again, we see a consistent pattern here. We can see on the left, the positive effect is improved in the active interventions, but not the maltodextrin. And the negative effect, they were all reduced, which is obviously desirable in all of the active interventions, but not the maltodextrin. Now, one thing that we were keen to look at is whether the Bifidobacterium responder status had anything to do with the responses. So when we do these clinical studies, we frequently see individuals that show a strong response. You feed people a prebiotic such as oligofructose, and you're going to get an improvement. You're going to get an increase in Bifidobacteria. We see this in study after study. But we also see some people that don't respond. So we're quite keen to explore whether the responders are having health benefits and not the non-responders. So if we look at the oligofructose data on the left there, you can see that there were 2 individuals there that didn't respond. They actually had lower levels of Bifidobacterium after intervention than before intervention. The maltodextrin, they're all over the place really. The blends, again, most people increased, but some didn't really increase very much, and 1 person decreased. With the human milk oligosaccharide, however, there seems to be an either-or situation. People are either responding or not responding. There seems to be a quite clear-cut responder, not responder situation with the human milk oligosaccharide in response to Bifidobacteria. In many ways, this isn't too surprising because the Bifidobacteria in the gut are highly specialized when it comes to their response to human milk oligosaccharides. Human milk oligosaccharides are a very, very diverse, heterogeneous bunch of carbohydrates. And there are specific Bifidobacteria that will respond to specific structures. So our hypothesis here is that if you happen to have the right bifidobacterial strains in your gut, then you will benefit from human milk oligosaccharides. If you don't, then you're not really going to see any benefit. When it comes to oligofructose and also inulin, we tend to see a much more general response. So most people will respond positively to those fructans. We don't know how widespread this effect is with the human milk oligosaccharides. And what we don't know at the moment really is whether we can correlate this with health. We did have a go at separating them, but the results are statistically challenged, to be honest. And this is an area that we're still investigating. So we are hoping to produce a follow-up paper where we look into this in a lot more detail. What we did is to divide them up into responders and non-responders. So in the purple, we have the non-responders. If you look at the BDI data, I think there it looks like the responders are responding more than the non-responders. but we need to follow this up with some hard statistical analysis. We can't really see very much with the STY-Y2, and the PANAS data are a little bit noisy too. But this is an area that we think is worthy of further investigation. So we're following up with a detailed analysis of our data, but I think this is something that really other researchers need to pick up on and look at. How do you respond to carbohydrate intervention, particularly human milk oligosaccharide intervention? I was hoping that we will be able to correlate the starting microbiome with the response to intervention. Unfortunately, we didn't have the data that would allow us to do that. So that's another area for the future, I think. So my conclusions, consumption of oligofructose and 2-fucosyllactose produced clinically significant improvements in mood. Some individuals did not respond to 2-fucosyllactose, others did, whereas most responded to the oligofructose. So where do we go from here? Well, we need to repeat with a larger cohort size, and we need to do plasma metabonomics. We originally started this study in students, but we were rather horrified when we discovered how many of our students are on antidepressants. So we had to broaden this out. It took quite a long time to get this study completed. We had to keep broadening out and bringing more people in. Really, we need a study with a much larger cohort size right from the outset. And we also need to not do it in the middle of a coronavirus pandemic so that we can actually do plasma metabonomics. I think as well, we need to look at interactions with antidepressants and anxiolytic medication. We don't really have any answer to this. We specifically excluded people on medication. We were expecting a modest effect that wouldn't compete with medication, but we were very, very surprised by the strength of the response that we saw. So this really does beg the question, what are the interactions between medication and antidepressants? I think we need to do a study looking at whether or not prebiotics have adjuvant effects with medication for mood. We need to use a better measure of sleep quality, I think. I was a little surprised we didn't get any consistent data. Everybody improved their sleep quality in every group. Could be a placebo effect, but I wonder whether or not we use the right tool. And I think important question for me as a scientist, can we predict responder status from the baseline microbiome? This could be quite important in the future because, as I said at the start, it's actually now quite easy to profile the microbiome. We can take fecal samples from people and fairly rapidly get a detailed picture of their microbiome. If we can predict from that who would respond, then we can maybe start talking about targeted interventions in people that need it most. So what can health professionals recommend? Well, food supplements such as oligofructose will not compete with medicines, I think. I think it's still the case that medication is medication and food supplements are food supplements. However, recommendation of oligofructose and 2-fucosyllactose for people with mild to moderate anxiety and depression clearly would be a good idea. And there's certainly, I think, no reason why recommendation of these materials for people who are on medication. I don't see why that would not be a good idea too. So I'm going to stop talking there and thank you very much for your attention and maybe answer some questions.
Thank you so much, Dr. Rastall. We have several questions that came in. So let's jump in with our first one. Reflecting on the study results, how do you see the recommendation of taking prebiotics to positively affect mood and how we recommend the correct levels of inulin and oligofructose to the public?
Yes, that's the most important question, really, isn't it? I think we certainly can recommend taking prebiotics to improve mood. I think we have convincing data really that improvement should happen. Most people responded, particularly to the oligofructose. Very, very few people didn't respond to it. In terms of dose, it's difficult to say. We haven't actually got, obviously, a dose response study here. That is a study that would be worth doing, but dose response studies are quite difficult to do. They're very, very expensive, take a long time. Not many people like doing them. For general well-being, daily intake of 3 grams of chicory root fibers can already support digestive health and improve gut microbiota. For the mental health benefits, I mean, we used 8 grams per day. So I think that really is the dose that we would have to say has got the data behind it.
Great. All right, next question. Inulin is sometimes added to foods. Is this as good as eating foods that are naturally rich in inulin? And at what dosages do we see health benefits?
Yes, I do get asked this quite a lot. Inulin is a natural food component, of course. It's present in many foods, but it's not present in many foods at high levels. And the foods that it's present at high levels in aren't generally ones that people eat large quantities of. So it's found naturally in things like onions, garlic, asparagus. Most people don't eat large quantities of those. Artichokes are probably one of the richest, and chicory, of course, which is where the commercial product comes from. But again, chicory and artichokes are not usually consumed in large quantities by most people. What we do find is supplementing foodstuffs with commercial inulins and oligofructose has a measurable effect. There are very few studies that have actually been conducted looking at natural composition. I can think of one where people were put on a diet rich in inulin based on natural products, and it did indeed show benefits. But the diet that these people were eating is not one I think that most people would want to stick to on a long term. So I think supplementation is a way of gaining the health benefits of these natural ingredients, but in a diet that people would find acceptable.
Fair enough. Fantastic. All right, next question. You mentioned the definition of a prebiotic. There's so many out there that are claiming to be, well, so many products, I should say, out there claiming to be prebiotics. How can the public identify proven prebiotics from all the rest?
Yes, well, the term prebiotic is often used very loosely. It's used very loosely. Not all ingredients labeled as prebiotics meet the ISAP definition, and I personally think the ISAP definition is the one that we should go with. It's a consensus definition by people who know what they're talking about. There is a tendency for people to propose their own definitions, mainly people who want to get various things classed as prebiotics that wouldn't normally meet the requirements. So I think really we need to be following their recommendations. Now, healthcare practitioners can look at the paper that was published to support that definition, the references in the slides there. But really, I think proven ones are the inulin, fructooligosaccharides, oligofructose, and galactooligosaccharides. They're the ones that have got the science behind them. Those companies that have manufactured those materials have invested in science, and they've done the studies. Most of the others are kind of piggybacking on those studies. The thought seems to be that if inulin causes these changes in the microbiome and that has a health benefit, if my plant carbohydrate causes those same kind of changes, that will also have the same health benefit. I don't think that's necessarily true. Firstly, most people that are doing that are not characterizing the gut microbiome with enough detail. So you don't know for certain you've got the same changes that you have with, for example, oligofructose. And in any case, to meet that definition of a prebiotic, you need to establish the health benefits in humans. So those are the products I think people need to look for.
Excellent. And just for our listeners' sake, ISAPP, ISAPP, is the International Scientific Association for Probiotics and Prebiotics. So we're going based on what they're classifying.
It is. It is.
Perfect. All right. This will be our last question, Dr. Rastall. What are the implications of the recent findings on prebiotics and mental health for dietitians and healthcare professionals?
Well, I think there's a new opportunity to support patients' mental health through nutrition. And I think dietitians obviously are very much on board with the idea of improving health through diet. So the more tools that they have at their disposal, the better, really. So I think prebiotics will become more regularized, to be honest, as treatment strategies. This study is, I think, the first of its kind, but there are other studies happening as well. There's another one at Reading I know of. There's other studies around the world that I'm aware of happening. So I think we will see more data coming out in this area. The more data we have, obviously, the greater the support for using prebiotics. I think one thing to bear in mind with the prebiotic intervention is that you're unlikely to do much harm. These are natural food components. So really the only side effect of consuming prebiotics is a tendency for increased intestinal gas production. And that is transient. It doesn't last long. It's usually modest and it goes away. No one in our study complained of significant issues. Some people said they felt a bit uncomfortable for a few days, but they got used to it. So, these are safe and I think could be recommended to most people.
Excellent. So a small price to pay for a pretty large health benefit.
Yes, I mean, we were. I said it several times. I'll say it again. We were really surprised at the magnitude of the health benefit. We were expecting much more modest responses than we saw.
Excellent. Well, that's all the time we have for questions. I want to thank you, Dr. Rastall, for sharing your expertise. This was a fascinating presentation. We had a lot of excitement about it in anticipation for your talk. So thanks so much for being here.
You're very welcome.
Your attendance certificates are now available to download. You can follow the instructions on the screen or refer to the final slide of the presentation handout, which will be linked in our listing on our site for more information on how to complete the evaluation and then access your certificate. So thanks again, everyone, for being here. Thank you, Dr. Rastall, and I wish everyone a great afternoon and a great Thank you for attending our webinar. Your attendance certificate is now available to download. To claim your credit for this activity, log on to ce.todaysdietitian.com and click on My Courses in the upper right-hand corner of your screen. Locate this activity's name in your pending activities list. Follow the steps to complete the required evaluation and download your certificate. When recording today's activity in your CDR activity log, please select Activity Type 741, CDR CPEU Prior Approved Enduring Activities. Sphere and competency selection are at your own discretion.